This is a production of Cornell University.
Thank you. Thank you for the introduction, and thanks for having me. As you can tell from the title, there won't be many plants in this talk. So I thought, put in at least one slide with plants. So these are plants that we actually have in the lab. We don't work on them, but we like to have them. And obviously, since you guys are plant scientists, you must know everything about plants. Does anyone know what this plant is? It's an interesting one. This is, yeah? Amorphophallus titanum, a corpse flower. So we have a bunch of those in the institute now. I have one in my office as well. They're tiny and cute as a baby. I don't ever want to see it actually flourish and build a flower, but for now it's fine. I'll make this a little louder, maybe that's better. So yeah, okay, it does work. Okay, so my lab focuses on some very basic and very simple observations in fundamental bacterial cell biology. That's basically what we do. And one of those observations is this one, which is when you put a bacterial cell, like an E. coli, on a beta-lactam antibiotic, and you can see that the cells basically digest themselves and disintegrate. And there's a lot of interesting biology behind this very, very simple process, and we do a lot with that. It also has some obvious biomedical relevance because the beta-lactam antibiotics that kill the cell effectively here are among the most widely prescribed antibiotics in the US. And most of you, especially if you have kids, have probably heard of amoxicillin. That's usually given against otitis media in the form of augmentin, where the active compound, or the main active compound, is amoxicillin. So one of the questions we ask is, what keeps the cell from digesting itself and exploding when there's no antibiotic around? Well, most cells have some sort of a cell wall that surrounds the entire cell and contains these pretty severe intracellular pressures that bacteria elaborate. So some bacteria actually have intracellular pressures that equal that of a bike tire. And they have this strong cage all around them that keeps it all together. And this cage, as you can already see, consists of these polymers that are cross-linked with each other. And these polymers are actually polysaccharide strands. And these cross-links are short peptide bonds between them. And we know quite a bit about how bacteria synthesize this whole cell envelope, this whole cell wall. It all starts in the cytoplasm, where some precursor molecules are produced. So these are disaccharides with a pentapeptide site stem, and those are then flipped to the outside of the cell with by a specialized translocase called MERJ in at least a model organism, E. coli. And then those precursors are picked up and woven into the cell wall by two main reactions. One of them is a glycosyltransferase, and that is taking these disaccharide subunits and connecting them with each other. So you get these longish strands of polysaccharide. The second reaction is transpeptidase activity, whereby the cell cross-links these neighboring strands by these short amino acid site stems. So first, there's some elongation of the material, polysaccharide chain formation, and those polysaccharide chains are then basically knitted together via these little peptide bonds. And you already noticed that this all starts with a single pathway, and the translocation is a single pathway, and then it diverges. So then all of a sudden we have two enzyme complexes, the LPO-APBPs and the rod complex with BPBPs, that engage in these activities, glycosyltransferase and transpeptidase. In the APBPs, which stands for class A penicillin binding protein, should already be clear what the target or what these things are useful for in terms of drug development. The class A penicillin binding proteins have both glycosyltransferase and transpeptidase activity in one protein. The only thing they need is, in gram-negative bacteria, an outer membrane localized activator. Without the activator, they're toast. On the other side, we have the rod complex, which is a bit simplified here. It's more than just this one protein. There's a lot of proteins that are in this complex, but the main ones are rod A, which possesses glycosyltransferase activity, and a BPBP, a class B penicillin binding protein, which only has transpeptidase activity. And of course, one of the big questions in the field is, why these two? right? Why do we have two very different enzyme complexes that do exactly the same thing? One hint towards what they're doing is, if you look at their localization, I hope you can see that. Maybe you can. So this is just a time lapse, a short time lapse of cells that express labeled components of these different complexes. If you look at MRAB MSF GFP, which is a member of the rod complex, you can see how these it assembles into these foci that sort of slowly rotate around the cell. It's a bit like a tabloid newspaper. I'm telling you what you're supposed to see here, right? You're supposed to see rotation, but this has actually all been quantified and it checks out. This is actually rotational movement around the cell. If you look at PBP1AM cherry, obviously an APBP, that there's no particular pattern to this. So basically, these just diffuse around in the cell. And if you look at these, this has been done in E. coli, in another model organism. If you look at these at the single molecule tracking level, you will see that they basically diffuse around a little bit, and then they stop for a while, and then they go on diffusing. So these things biochemically do exactly the same thing, but they localize in very separate ways, and they seem to be very separate complexes. And I think so far, all of this is sort of known and to a degree boring because it's known. So what we're really interested in is how all of this leads to growth and how the bacterium deals with an interesting paradox. The paradox is this. You have this cage that contains a huge intracellular pressure. If you take away the cage, the bacterium dies. And yet, this has to be really, really flexible because bacteria have to grow, for example. Most bacteria, when I say bacteria, I typically mean model organisms. There's, of course, a variety of different strategies. But the model organisms, they start off as a short cell. They elongate, and then in the process, they have to deposit new cell wall material in the side wall. Then they make a new cell wall in the middle of the cell, and then they have to cut that in two, so you end up with two equally sized daughter cells. And you can tell from this that just cell wall synthesis it's really not enough to explain this. Here's the reason. So imagine you have these pathways, precursors come in, they get assembled into the cell wall. All you end up doing is you increase the thickness of your cell wall, if this is everything that's going on, right? And that doesn't help actually elongate anything. So unsurprisingly, there's a whole different side to this, which is a variety of enzymes that can actually break the cell wall. There are four main families. They're often hugely redundant in bacteria. The Amidases that cut away the peptide site stem from these polysaccharide strands, the lytic transglycosylases that cut within the polysaccharide strands, and the endopeptidases, about which I'm going to tell you a lot today, that basically cut within these crosslinks. These are often collectively referred to as autolysins, because if they are uncontrolled, they can lead to cell lysis, and they're able to cut the cell wall sacculus in various positions. We know that these exist, of course, because of an experiment I've already shown you. If we use a beta-lactam antibiotic, what we do is we inhibit the penicillin binding proteins, which of course bind penicillin and are inactivated as a consequence. And now all of a sudden, the action of these autolysins becomes apparent. Here's another example of where we see that. So we inhibit cell wall synthesis. This is meropenem, it's also beta-lactam antibiotic. And your average E. coli cell will lyse and die. And my model organism, Vibrio cholerae, does something really cool. It doesn't lyse and die. Instead, it will crack open, spill out its guts, and then form these spheroplast-like structures. And what happens in these two images, or in these time lapses, is exactly the same, right? The cell wall gets degraded by autolysins, but here you still have a cell, and here you don't. And we're cell biologists. We need a cell. So this is a perfect system to actually study this process, which is not possible in E. coli, where everything rapidly falls apart and the cell gets destroyed. As a little side note, we've noticed that this is also an interesting, or potentially an interesting mechanism by which bacteria can resist treatment with antibiotics in the clinic. In collaboration with Lars Westblade from Weill Cornell, we obtained some clinical isolates of gram-negative bacteria, exposed them to carbapenem antibiotics, and we found that many of them, Enterobacter cloacae, Haemophilus influenzae, Klebsiella aerogenes, and pneumoniae, also form these spheroplasts. And if you then wash away the antibiotic, these spheroplasts actually rapidly come back to life and form a microcolony again. So this might allow them in the clinic to actually survive antibiotic exposure. So we think this is a mechanism of antibiotic tolerance as well. Now I want to point out Haemophilus influenzae, since I already brought up otitis media. This is one of the causative agents of otitis media. And maybe this is the reason why otitis media actually is sometimes difficult to treat. Not because of resistance, but simply because Haemophilus hangs in there as a spheroplast. After a course of 10 days, you stop taking the antibiotic and it will start growing again. So what is the normal function of all these lytic enzymes? It seems kind of like playing with fire, right? Why do bacteria have all of these around? Well, they are important, as I already suggested, in various growth processes. For example, we know this very well. The amidases that cut the peptide side stems here are important for daughter cell separation. And this is sort of known from E. coli as well, from a variety of organisms, so it seems to be very well conserved. If you delete amidases, in Vibrio it's only one, instead of these nicely separated daughter cells, you basically get these long cell chains. The cells are fine, they grow fine, but they can't separate anymore, so they stick together. So seemingly the septal PG that is produced in these cells can't be cleaved anymore. And more recently, a grad student in my lab figured out that you need a second factor. It's not just the amidases, you also need lytic transglycosylases to cut the septum. Because if you knock out all the lytic transglycosylases, or two that matter in this case, you also get these long cell chains. So we think that the septum looks something like this, where you have a shared peptidoglycan cell wall between these two cells. And the amidases, of course, can do most of the work, but the amidases cut here between the peptide side stem and the polysaccharide strand. So what if you have a polysaccharide strand that stretches from one cell to another? Well, then you need the lytic transglycosylases, and these sort of come together to resolve the septum. I think, most important autolysins, however, are the endopeptidases. So these are all zinc-dependent enzymes, which will become important later, and they're important for cell elongation. And we know this because we can knock them out. There are two that matter. They're synthetically lethal with each other, so we can knock one out, keep the other under control of an inducible promoter. As long as it's expressed, you can see here in time lapse, the cells elongate, divide, and do what they're supposed to. If we deplete this endopeptidase, so now it's effectively an endopeptidase minus mutant, you don't get elongation anymore at some point. You can see that division actually proceeds, and you end up with these short stubby cells. We can quantify that, so elongation rate goes sharply down as we deplete ShyA, and all you need to do is express ShyA back in, and we get wild-type elongation rates. So the endopeptidases are important for elongation. Why? Well, we think it's something like this. However, I have to warn you, this complex that's shown here has never been actually proven to exist. And we don't think it is actually a complex. So let's take this as sort of a metaphorical complex of activities rather than of proteins. Because the activities that have to come together to enable cell elongation are something that cuts out an old strand of peptidoglycan and something that fills in two new strands of peptidoglycan, 3 new strands of peptidoglycan. It's 3 because that's how the stoichiometry works out. So you can actually measure turnover in cells, and it's something like 3 to 1. And the endopeptidases would cut here, right, in these peptide crosslinks. And that's how you get cell expansion, because you can imagine if you cut out these cables and then supply some more cables here, you get lateral movement, and that's how the whole cell elongates. And we think that that's what the endopeptidases do. It's a little bit more complicated than that, though, because we still don't know what the relationship between endopeptidase activity and synthesis is. Do you actually need endopeptidases in order to synthesize anything, or can they actually be uncoupled? And we have some indication that they can be uncoupled, because now we look at a strain that's deleted in all endopeptidases. There are more than just those 2. So now we took all of them out. And if you follow a very, very simple experiment, you just follow the mass increase in these cells. And without any endopeptidases, there's still a significant mass increase. They don't seem to care all that much, right? If you look at these cells, you will notice that they just become grotesquely enlarged, but they still enlarge. So they still do something. They still seem to incorporate peptidoglycan cell wall material. Of course, with endopeptidase, you get normal cell growth. We can measure that. We have indeed more cell wall material. And we can also visualize the cell wall material with a fluorescent cell wall stain. And indeed, these things still incorporate cell wall material, but it looks aberrant. So normally you would expect something like that, where the cell is evenly fluorescently stained. But in these endopeptidase depletion mutants, you just have this accumulation of bright spots. And this is one of these rare moments where I finally have some tools at my disposal that I'm always jealous of with the eukaryotic world, because the eukaryotic cell biologists seem to have inhibitors for everything, right? So you see a talk and they implicate some pathway in something, so they just buy the inhibitor and can test it easily. Finally, we can do that too, because there are very specific inhibitors for APVPs and for the rod complex. And this allows us to ask the question, this mass increase that we see without endopeptidases, what is that actually mediated by one of these systems or both of them, or which one is more important? And that gives us important information about the relationship between these different complexes and the cleavage activity by endopeptidases. And very simple result, we inhibit the APVPs, deplete endopeptidases, and all of a sudden there's no more mass increase. And if we inhibit the other system, the rod system, you still get the same mass increase, meaning that whatever aberrant peptidoglycan incorporation that you just saw on the previous slide is all mediated by the APVPs. Or in other words, without endopeptidases, the rod system is completely useless. And this sort of explains some of our phenotypes with the antibiotics, right? So here's again, this is effectively a wild-type strain because it still expresses one endopeptidase in Vibrio cholerae. You can see what I showed you before. You get this spheroplast formation. And if we take away all the endopeptidases and repeat this, now we don't get the mass increase anymore. And instead, the cells try to elongate a little bit and then lyse and die. So now Vibrio cholerae looks a lot like E. coli. Meaning that for the spheroplast formation, we actually need endopeptidases to degrade the cell wall in a controlled manner. And we basically already knew this since the 1980s, that endopeptidases and also lytic transglycosylases are important for cell wall degradation in the presence of beta-lactam antibiotics. It's just nice to see that in a time lapse sometimes. So why am I telling you all of this? I want you to know that endopeptidases are major contributors to antibiotic-mediated cell wall degradation, and that's mainly why we study them, because we want to figure out how they're regulated. I also would like you to remember that spheroplast formation, the sphere formation phenotype, is a readout for toxic endopeptidase activity. If you have too much endopeptidases, or if they're imbalanced because you inhibited cell wall synthesis, you get spheroblasts. And now the questions that we're asking in the lab is, how is endopeptidase activity regulated under normal growth conditions when we have no antibiotics? How does the cell actually keep all of this in check? And I'm going to tell you two stories about this today. One is endopeptidase regulation is an adaptation. So this is genetic regulation of endopeptidases. This was just published in MBio. And then the second story is about a news story that we hope to get into bioRxiv next week or so, about a conformational switch that promotes endopeptidase activity. So this is post-translational endopeptidase activity regulation. So the first one, as ever so often in science, goes back to an extremely simple observation. You've noticed by now that Vibrio cholerae has a lot of these endopeptidases around, right? And I told you that two matter, ShyA and ShyC, but there's also a third one that basically looks exactly like ShyA and ShyC. It's called ShyB. Has the same domain organization, seems to be doing the same thing. However, it wasn't able to sustain growth. As you saw in the time lapses, we can't actually delete ShyA and ShyC, right? So ShyB is useless. We then noticed that it's not actually even expressed. You put these cells on LB. This is a reporter construct for promoter activity. Blue means promoter is on, white means it's off. And you can see that ShyA and ShyC are constitutively expressed, and ShyB is completely off. So what Shannon did, my grad student, is something very simple. She took this reporter construct, mutagenized it with transposons, put it on a giant plate, and looked for mutants that now spontaneously induced the promoter. And this is something that's actually quite amazing for a grad student. She did this basically within a month of joining my lab, and immediately got something out of it, and immediately ran with it, and within a year, or even less, had a paper. Science doesn't always happen like that, but here it did. So she did this, and you can see in the sea of white, there are some blue colonies here. And here is a zoom in. So some of these colonies turned blue, which means we found a mutant that now induces this promoter. And you can see some are actually a little more blue than others. And I was very excited because I thought we got the master regulator of endopeptidase activity. Instead, we got something called Zur. Zur is weird. So Zur is a zinc starvation response gene. I'll tell you in the next slide what it really does. And the other hits were in an operon called ZNU A, B, and C. And those were all the hits. So we drove this to saturation. This is all we got. What does all this mean? Well, Zur is, as I mentioned, a zinc starvation response. So Zur is a repressor that sits on certain promoters controlling zinc starvation response genes. If zinc becomes limiting, Zur release repression and the zinc starvation response genes are turned on. Part of this whole repertoire of zinc starvation response is something very intuitive, zinc importers, right? You don't have zinc, you want zinc, you pump in zinc. And there are two of them. And these, if you may have noticed in the last slide, are our other hits. So the other hits are in this primary zinc transporter. The difference between those two, by the way, is this is a high affinity transporter. So this is useful for for low zinc concentrations. And this is a low affinity zinc importer. Other zinc starvation response genes include replacement proteins for example, ribosomal proteins that typically require zinc for stability. So this is all basically ensures that the cell can sustain viability without much zinc around. So it seemed like the Zur regulon also contained a cell wall remodeling enzyme, which was unheard of and kind of weird. So first we went ahead and actually validated that that's true. So we looked at the promoter, the ShyB promoter. It did some 5' raise, so we know where the transcription start site is. And turns out the -10 region has an overlapping, beautiful, well-conserved Zur box. And we would have never looked for a Zur box, a Zur binding site ahead of this if it hadn't been for the screen, right? So we validated binding of the promoter with an EMSA assay. So this just shows that that Zur indeed binds to the ShyB promoter. So it seems like it all checks out, and ShyB is a bona fide Zur controlled gene. Biochemistry is fine, but I kind of like genetics. So we also wanted to see that checks out genetically. We noticed that in certain minimal media, like M9, cells seem to be starved for zinc, and they turn on Zur. And that's why the ShyB promoter, this again, the ShyB promoter construct, is actually on an M9 minimal medium plate. If you work with bacteria, that's something you should probably remember, because I don't think people realize the starvation conditions that they're exposed to in M9. So in M9, the ShyB promoter is on, suggesting zinc starvation. We add zinc, it turns off. Beautiful. It doesn't turn off anymore if we delete Zur, because Zur is obviously the repressor. If you don't have it anymore, you can add as much zinc as you want. You won't repress the promoter anymore because its repressor is gone. And then delta ZNU ABC in the zinc importer mutant that we created, you can still shut down zinc promoter activity of ShyB, suggesting that the second zinc transporter, that's sort of one of the tangents that came out of that, seems to be functional. If you just add enough zinc, it'll be able to pump it in and repress this promoter at some point. And we can also quantify all this. So again, in the wild type, you can effectively shut down ShyB promoter activity by addition of zinc. You cannot do that in delta Zur, of course, because the repressor is gone. And in delta ZNU ABC, now relying on zinc import by this low affinity transporter, the kinetics are a bit less steep than for the wild type. So then the question was, is ShyB really functioning as an endopeptidase here, or maybe does it something else? Well, the summary of this is, yes, it's an endopeptidase. At least it can substitute for ShyABC in vivo. So if we look at these mutants, a lot of control strains, but the ones that matter are the delta ShyABC, delta ShyC. So these are all conditional. They have the other endopeptidase under inducible control, but it doesn't matter here right now. So delta ShyAB is fine because it still has ShyC. Delta ShyAC is not fine in a normal growth medium because ShyB is simply repressed by Zur. However, if we add a zinc chelator, things turn around. So now we take away all the zinc. ShyAB, delta ShyAB can't grow anymore, suggesting that actually ShyC is inactivated somehow by the absence of zinc. But now the ShyAC mutant can grow because we chelate zinc, we induce Zur, ShyB gets induced, and apparently ShyB is enough to take over and can do the job and can sustain growth. Genetics is fine, but I also like biochemistry. So let's look at this in another way. We wanted to test whether ShyB is an endopeptidase indeed. And now I think it slowly, maybe for you all, comes together that we're dealing with zinc-dependent enzymes The zinc starvation response usually includes enzymes that replace enzymes that typically require zinc. Might this be something similar for ShyB? Might ShyB actually be an endopeptidase that doesn't require zinc? Well, we tested that using biochemistry. We can purify endopeptidases, then take cell wall sacculi and digest them in vitro. And there are several reaction products here. I'll walk you through them. The most important one is the soluble fraction. So these purified sacculi can be sedimented with ultracentrifugation. But if endopeptidases start cutting away, they will cut the crosslinks, right? So they will liberate single strands of peptidoglycan, and that will end up in solution, and you can measure that with UPLC. And then we add an enzyme called muramidase. It's not really important what it does, but it creates basically diagnostic cell wall fragments that allow us to gauge the activity, the specific activity of these enzymes. So this is what this would look like. Focus on the middle part at first. So the first thing you should notice is that ShyA, B, and C can all digest something, can cut something out of the sacculi, right? So there's something in the soluble fraction here and not in the control or an active site mutant of ShyB. You may also notice that it looks a little different between ShyA and ShyB. And we don't actually understand that. So it seems to have slightly different activity. But in any case, all of them cut something. And when we then digest this with a muramidase, revealing these diagnostic peaks, we notice that this reveals for all three endopeptidase a D-D endopeptidase activity. So they're indeed equal in terms of that. The specific activity seems to be a little different, but all of these have the same cutting specificity. All of them are the same type of endopeptidase. What we can then do is we take the area under the peak here and quantify reaction kinetics. And that's plotted here. And then we repeat this experiment in the presence of EDTA and a chelator of zinc, asking the question, can these endopeptidases actually remain active without zinc, or without metals in this case, because it's not specific to zinc. And for ShyA and ShyC, the answer is no. So if you add increasing concentrations of EDTA to these reactions, activity gets pretty rapidly shut down. For ShyC earlier than for ShyA. But if we repeat the same thing with ShyB, it remains completely active even in the presence of very high EDTA concentrations. So ShyB seems to be able to function either in the absence of zinc or it just has an extremely high affinity to zinc. We don't know which one it is yet. We're working on that. So we still thought it's kind of weird, right? Because the zinc starvation response, like most metal starvation responses, is very well understood. So why hasn't anyone stumbled across an endopeptidase and all that and made the connection to cell wall remodeling? Well, we think it's been overlooked because ShyB, we're kind of lucky because our model organism probably has the only endopeptidase, the only one we're aware of, that has a Zur box in its promoter. So it's more recognizable that way. But there are a lot of bacteria that have something similar, that actually have other Zur-regulated genes, like ZNU-ABC, the transporter, and just downstream of that is actually an endopeptidase that's homologous to ShyB. And all of these organisms have that, suggesting that in zinc starvation, basically ZNU-A and this endopeptidase are co-transcribed. And there's some evidence, if you dig very deep into papers and their supplemental data where people did RNA-seq analyses, for Yersinia pestis and for E. coli, that's actually been shown that zinc starvation induces this endopeptidase. And this is significant because zinc starvation is something that cells encounter frequently in the human host. So this is something they have to deal with, and perhaps ShyB is actually one mechanism of how to remain viable upon encountering host pressures like zinc sequestration. And another little thing, Eric Scars on many of these papers, the same day that this paper came out, his paper came out. We didn't know about each other, but basically showing exactly the same thing in a different organism. And that's really nice if you get validated that way, that someone basically not actually reproduces your data, but shows exactly the same in a very different organism. So this really suggests that this might be widespread and important for pathogenesis. All right, so that's basically the genetic regulation part of the talk. I want to move on to something that we're really excited about right now, which is a conformational switch story. So I've always been puzzled by one thing. I've already told you that all these autolysins are potentially really, really dangerous, right? If their activity goes out of whack, they destroy the cell digests itself and dies. However, you can clone these into fairly high copy number plasmid and overexpress these in various bacteria. This has been shown in this case as Vibrio cholerae, but the E. coli crowd sees the same thing, and absolutely nothing happens. So it seems like you can really overexpress dangerous lytic enzymes, and the cell doesn't care. We made it care in an extremely contrived situation where we take ShyC, which is typically in the inner membrane, and we basically cut off its transmembrane segment. And then if we overexpress it deep, deep, deep into this phase, into the stationary phase, at some point we see some sphere formation, which I've told you is indicative of toxic endopeptidase activity. So we can make these work, suggesting that they're not simply dead here, but we really have to blast it out of the water. So this all suggests that endopeptidase activity in vivo is actually highly regulated. However, what's puzzling is, at the same time, you can purify these enzymes, and they're highly active. Actually, within minutes, you can purify saculi with purified enzymes, like I showed you 2 or 3 slides ago, and they're totally active. So that is kind of bizarre, because the lytic enzymes, the autolysins that we know about, or the regulation of which we know about, they're all produced in an inactive state, and they're activated by, well, specific activator proteins. So if you purify them, they're inactive unless you add the activator protein. That doesn't seem to be the case for the endopeptidases, because they're highly active. So it could be that there's some sort of a negative regulator, but that you would overcome at some point, at least here during rapid growth. So it's kind of puzzling, right? And I think we understand this now due to a wonderful collaboration with these individuals. So this is Mark Saper at the University of Michigan, Yujin Mao and his grad student Ellen here at the Weil Institute. And in collaboration with these 2 labs, we obtained crystal structures of endopeptidases. And this has really, to me, has really increased my appreciation for crystallography. I always thought crystallography was sort of the end point, right? You show something genetically, and then you prove it with a crystal structure. I didn't realize it can also be hypothesis generating, and that was true in this case. So the first structure was boring. The first structure is something that you will see in the published literature. People have actually crystallized endopeptidases, and they always see something like this. I'll just orient you here. So this is the N-terminus. This would be domain 1. Then there's a domain 2 and a domain 3 in the C-terminal helix. Domain 3 has the active site. This gray ball here, that's the zinc. And this is where peptidoglycan would enter and then be cut. The second structure was very exciting. We don't quite understand yet why we got these 2 structures, by the way. These were differentially tagged. This was tagged N-terminally, the C-terminally. We think that maybe the C-terminal tag might have destabilized this whole thing, so it opens up a little bit. And you can immediately see that this, if you know something about the mechanism of action of these enzymes, that this active site looks rather closed. There are these loops here that are occluded, and everything's kind of closed up. And in this structure, all of this is a bit more open. So the immediate obvious hypothesis here is that this is actually an active conformation, and this is an inactive conformation of an endopeptidase. And what happens in vivo is that some dramatic conformational rearrangements activate these endopeptidases where needed. I want to note here that it's probably in vivo not this dramatic. Our crystallography conditions might have actually favored this. So it's probably something in between, some slight opening, and that should be enough to actually increase activity. So this is, if we look at the active site in the open conformation versus the closed, we modeled in a peptidoglycan fragment here. So this would be the peptide crosslink, and the polysaccharide strands would be here. And you can see in the open conformation, indeed, the active site is nice and wide open. So this structure would actually fit in here. And in the closed conformation, it would not. It would be occluded by, for example, this residue, but also by this gigantic domain 1 that is not drawn here, that would basically reach into the active site and close it up. So we can test that now, of course, with relevant mutants. I already told you cells don't care whether they overexpress ShyA or not. So this is ShyA on a multi-copy number plasmid, either uninduced or induced, and there's not much of a difference between those 2 conditions. However, if we delete domain 1, which we predict to inhibit function, we get something like this. We get much smaller colonies. They kind of look sickly. And I will not ask you to believe that. This is, again, tabloid, right? I'm telling you what to see here. This is just for the Vibrio aficionados. You know that this is wrong. But if we actually look at these cells under the microscope, we see some appreciable differences between either active site mutant overexpression, wild type overexpression. and this. You get sphere formation, a high degree of sphere formation in some lyses. So this strongly suggested that domain 1 is indeed inhibitory. If you take it away, you get a more active enzyme. Deleting 1/3 of the protein is kind of a crude machete type method, of course, so we looked a little closer. What other mutations might favor this open conformation? And we looked at the interface between domain 1 and domain 3 that's supposed to be inhibitory. And there are several residues here that, that are predicted to mediate some sort of an agglutination of these two domains. And one particular one that's my favorite is these two leucines that engage in some sort of a hydrophobic interaction. And they're embedded in a sea of acidic and basic residues respectively, depending on the two domains. So we thought if we mutate one of them to something charged, maybe we get higher toxicity, for example, to a lysine. And we did that, and now cells get completely obliterated by this mutant protein. And this is quite amazing because this is a mutation very far away from the active site in domain 1 that increases toxicity to a degree where you actually get a plating defect. So we were very excited about that. So now we introduced a basic residue instead of this leucine here in domain 1. Oh yeah, and these of course also form spheres and check out that way. So now we can reverse that and actually then mutate the other residue to an aspartic acid. So now this should be glued back together, right? And indeed it is, or at least we think, because we lose toxicity of this mutant again. And if we look at these cells, they look totally happy and healthy. So this strongly suggests that indeed these two domains interact with each other, and if we open them up a little bit, the enzyme becomes more active. We did this biochemically as well. Again, it's always nice to have a combination of genetics and biochemistry. We purified these mutants, and again, in collaboration with Felipe Cava in Sweden, did these activity assays, and we saw something that's a little bit strange. We still don't understand that, but first, what we do understand is that the wild type, and we used the closed conformation, by the way, the construct that yielded the closed conformation, the crystal structures here, takes hours, basically an overnight, until it finally digests cell wall material to completion. If we look at the L109K mutant, the hyperactive one, within 10 minutes, basically everything's digested. Intriguingly, the L109K L344D double mutant, which was non-toxic in vivo, was still pretty toxic or pretty active in vitro, right? There is a significant difference between these two, again, suggesting that indeed this double mutation reduces toxicity of L109K, but it's still pretty active. And we think it has something to do with the specific environment in the periplasm of the bacterium, molecular crowding, for example, that is better at stabilizing this closed conformation. And in vitro, it sort of artificially opens up. Here's another mutant. I just want to show that we've tested a variety of mutants along this interface between domain 1 and domain 3, and they all basically cause some degree of toxicity. This is the 112E, you get sphere formation, a little bit of a plating defect, or rather colony morphology defect. And one more thing I want to mention is that the phenotype of overexpression of endopeptidases is not actually just destruction of the cell wall. We noticed when we used our cell wall stain that some of these, especially the delta domain 1 mutant, actually led to spheroplasts that have a pretty decent cell wall. So they're fully surrounded by a cell wall. And this is reminiscent of our inhibitors. If you inhibit the rod system, this is basically what you get because the APBPs can still do the job. If you inhibit the APBPs, everything falls apart, and that's closer to something like the L109K mutant. This is significant because there is this old, not so old actually, this realization that it seems like endopeptidases actually favor cell wall synthesis activity. So this is an assay where people checked cell wall synthesis activity by the rod system and the APBP system. And they noticed that if you overexpress an endopeptidase, like MEP-S in E. coli, you get an increase in cell wall synthesis for some reason, which is a bit strange. So there seems to be some connection, and I told you we're interested in the specific connections between either the rod or the APBP system with the endopeptidases. There seems to be a specific connection between ShyA, or an endopeptidase, and the APBP system. And this might actually explain why we see this conformational switch, we think. So this now gets into the very speculative area of my talk, because we did a pull down, a SciLag, looking for ShyA interaction partners. The hypothesis behind this is that, let's say there is a protein factor that favors the open conformation, or that induces open conformation in ShyA. We should be able to find it by pulling it down. And what we get is a lot of things, but what is the overlap between the two endopeptidases, ShyA and ShyC, is LPOB. You might remember LPOB from this diagram. LPOB is an activator of APBPs. So this does suggest some connection between ShyA and cell wall synthesis. And the simple working hypothesis here is that ShyA only gets activated once it interacts with the cell wall synthesis complex, and that's how you regulate degradation and insertion of new material without loss of structural integrity. We don't have much to go on yet, but we can use this horrible phenotype from a few slides ago to at least run some preliminary tests on it. So this ShyC, devoid of its transmembrane signal, induces sphere formation at some point. So maybe the reason it does that is because if we overexpress it freely floating in the periplasm, it has more of a chance to interact with its activator, LPOB. Which means that we repeat this experiment in delta LPOB, we shouldn't see sphere formation anymore, and that's exactly what happens. So that tentatively to me suggests indeed that LPOB might be an activator of endopeptidase activity anymore as well, and this would be a very simple model then, that you get this conformational switch, and the interaction with the cell wall synthesis machinery stabilizes that and activates endopeptidases. Since I work on a non-standard, non-model organism, Vibrio cholerae, we always worry about convincing the E. coli world. I don't know how it is in plant sciences, it's probably similar. There are specific model organisms that people follow. And if you work on something that's not found in these model organisms, people don't care as much. So we checked, does this actually happen in other organisms as well? So we pulled out some endopeptidases from varying organisms. How am I doing on time? Fine, right? 5 minutes? I'll go through this fast. So we pulled out some endopeptidases from various organisms that all sort of look the same. They all have the same domain organization. And then we created mutants that are expected to mimic this activating phenotype. For example, for MEP-M in E. coli, we can cut off domain 1, get full toxicity. For a Neisseria gonorrhea enzyme, we can create a point mutant in this interaction interface between domain 1 and domain 3, and again, we get higher toxicity. So it seems like pretty widespread, this mechanism of activation, and not just a weird curiosity of the Vibrio world. And so as a summary, I've shown you that endopeptidases are important for cell wall degradation after antibiotic exposure. They're only required for the rod system, but can enhance APBP function, and they're regulated in various ways to ensure that the cell coordinates cell wall synthesis and degradation. So with that, I'd like to thank you for your attention, and I'll take any questions you might have.
You ever find suppressors of the human ShyA?
Yes, but they're probably not very interesting at this point because they're usually in our expression system. So we get mutant in our expression system. I've thought about setting this up in a way to get something more informative. For example, you could have several plasmids that overexpress the toxic ShyA variant. So I think those would be very interesting, but yeah, we haven't looked at that yet.
In zinc starvation conditions, would the ShyB be a limiting factor for proliferation or for survival? Maybe just for survival?
Curiously not. So this is a bit weird. It seems like ShyA, even though it can be effectively shut down if you remove its zinc, in vivo it actually doesn't seem that different from ShyB. ShyC is clearly the one that under zinc starvation conditions doesn't work anymore. And I always say that these are synthetically lethal and functionally redundant. They might not be entirely functionally redundant in an actual in vivo situation. ShyB is not necessary for colonization of mice, but that's not a real disease model. And we would have to probably go into humans to actually test this hypothesis.
Any other questions? I have a question.
Yeah.
So why is E. coli exposed to antibiotics bursting and not spheroplasty? Is it missing some of the endopeptidase activity or machinery?
So the simple answer is I don't know. And it's, of course, a question we're really, really interested in. We've tried several things. Curiously, if you overexpress ShyA in E. coli, it helps it a little bit. So it will actually form spheres in response, suggesting that you actually need a certain type of degradation to survive this. And I feel like it might be something like a balloon. If you just pop it in one spot, it'll burst. But if you maybe cut it a little faster or in a different way, it just slowly unravels. And then you don't get this pressure release along a tiny speck of cell envelope surface, but rather like the slow release of pressure that enables them to stay alive. But that's not the only thing. So there must be something else. The outer membrane might be more fortified in Vibrio cholerae, with some indications that that actually might be the case, but nothing definitive yet. Yeah.
I have kind of an odd question, I guess, and that is when you're using OD600 to monitor the cell mass or whatever of these bacteria, I mean, aren't there sort of effects of the morphology of the bacterium in terms of the optimal density? I mean, I would think that, you know, if you do plate counts, for example, do you see that there's not artifacts resulting from OD600 itself?
So there are two things to this. First of all, you're absolutely right. OD600 readings are not reliable with these weird morphologies. But when you say, or when I say unreliable, that means there's no connection anymore, no true connection between CFU and cell mass, right? If you look at mutants like that, like depletion, where they actually die because they need endopeptidases, then of course the connection between CFU, between live cells and cell mass is completely disrupted. So basically the endopeptidase mutants die when we deplete endopeptidases, but they still increase in mass. And they die, I think, because they increase in diameter too much that they can't go through cell division anymore. So we've done some experiments where we let them recover. If they're under a certain diameter, they can still recover. Otherwise, they can't.
Any other questions? Well, if there are no questions, let's thank Toby again.
Thank you.
This has been a production of Cornell University, on the web at cornell.edu.